chainsaw movies and hockey mask villains, if you want some real horror, take a look at this. The living, blood-sucking tick. This is what Europeans call a wood tick. It's a cousin of the deer tick, and they both spread Lyme disease. They're both about the size of a sesame seed. It has a mouth that looks like two saws on the side of a barbed sword. That sword is called a hypostome. Scientists who study Lyme disease wanted to find out how this tick, with that mouth, manages to both cut through the skin and then to hang on for the days that it takes to get a full blood meal. They took video of the ticks biting into the ears of hairless mice. Now the mice were already dead. They were sacrificed for the experiment. What they saw was that once the tick picks its spot, it uses those saws to cut into the skin, moving back and forth, doing something that looks a little bit like the breaststroke. And that pushes the hypostome in and pulls the skin back over. Once the tick is solidly embedded in the skin, it doesn't have to expend any energy, so it can relax and drink its fill. There's no practical use for this knowledge yet, but it doesn't hurt to know your enemy.
Between Ixodes cookii and Ixodes scapularis, and more importantly, this species determination is in consensus with that which we determined using 28S. So using these genetic analyses, we have double assurance of the species of each unknown tick. And of the 10 ticks analyzed, exactly 5 were Ixodes cookii and 5 were Ixodes scapularis. So now knowing the species of each previously unknown tick, we can go back and compare their morphology to that of their respective species average. This graph is showing you the ratio of pulp length to pulp width. So as you'll recall, Ixodes scapularis is expected to have longer, narrower palps, so an overall larger ratio, where Ixodes cookii is supposed to have shorter, wider palps, so an overall smaller ratio. And when we look at the respective species average, this is what we see. And so if we were expecting to see little or no phenotypic diversity, we would guess that any individual of these groups would fall within a relatively close range of those species averages. This is not, however, what we see when we look at the morphologically ambiguous ticks. Here, once again, are the species averages, but the individual values are markedly variable. And more importantly, there's overlap between the two species. Here's a nice example of what I've just shown you. We have Ixodes cookii on the left and Ixodes scapularis on the right. And if you look at the mouth parts in both of these specimens, they're virtually indistinguishable. And this ambiguity is not limited to the mouth parts. We also see this in the shape of the sputum. Here, once again, Ixodes scapularis on the left has a sputum which is comparably angular to that of Ixodes cookii. Lastly, I looked at the color of the legs using cumulative red, green, and blue values as a proxy, and essentially found the exact same thing, a lot of variation and overlap between the two species. And looking at this morphologically ambiguous tick, we can see that contrary to popular belief, Ixodes scapularis, also known as the black-legged tick, can indeed have brown legs. So in conclusion, both Ixodes cookii and Ixodes scapularis are capable of far more phenotypic diversity than has been previously appreciated or documented. And this obviously compromises surveillance efforts and has potentially resulted in the underestimation of the prevalence of Lyme-competent ticks in our region. Knowing this, we cannot move forward and continue to define the species based on morphological characteristics, such as sputum shape, mouth parts, and leg color. And more importantly, there's one other significant implication. But its sputum is very angular like that. And so we ask ourselves, what species is this? Well, we actually don't know, but because the difference between Ixodes scapularis and Ixodes cookii is the difference between a recognized Lyme disease vector and one that's overlooked entirely, it may not surprise you that morphologically ambiguous ticks such as this have been the cause of some concern. And this is what's prompted my research. There were two hypotheses proposed to sort of explain this observation of morphological ambiguity. And the first was that close proximity and relation between Ixodes scapularis and Ixodes cookii had actually given rise to some Ixodes hybrid. Our second hypothesis was that one or both of these species are actually capable of more phenotypic diversity than we previously alter their phenotype without changing their species. And sometimes this can be an adaptive response, and you're well versed in this now, but it's sometimes called phenotypic plasticity. And so we often see this in insects, particularly in butterflies, who can change their wing patterns drastically based on seasonal variations. So the individuals in each column are actually the exact same species, even though they look very different. So how did I test these hypotheses? Well, part one of my project was a morphometric analysis, and this was to quantify species-specific characteristics, as well as to assess the possibility of intermediate and mixed morphologies. Part two of my research was to do a genetic analysis, so actually look at the tick's DNA in order to figure out what the species were of our unknown ticks, as well as to assess the possibility of hybridization. So for my morphometric analysis, I used imaging software to measure several characteristics. I first looked at the length of the palps, the width, the hypostome length, the leg color, and the shape of the sputum. And you'll remember these are all expected to be species-defining characteristics. For my genetic analyses, I randomly chose 10 ticks from each group. So I chose 10 which we thought were Exodus scapularis, 10 which we believed to be Exodus cookii, and 10 of these little guys with morphological ambiguity whose species was unknown. The first gene I looked at was the 28S ribosomal RNA gene. Now, between Exodus cookii and Exodus scapularis, the sequences for which are shown here one on top of the other, there are 7 known nucleotide differences between these two species. So if we were looking for hybridization, we could look for mixed combinations of these markers. So I amplified this gene in all of the samples using polymerase chain reaction, and this was so that I could compare the individual's tick DNA to that of the published sequences shown here. So I designed forward and reverse primers which attach at either end of the gene and transcribe or copy the gene in either direction so that we end up with multiple gene copies. I then take this amplified gene product and I send it away for sequencing. Now I did this same process with one other gene, cytochrome oxidase subunit 1, which is a mitochondrial gene. And this just means that it's internally inherited. So by looking at both genes, we have double assurance of the species of each unknown tick. And these are my results. So we're looking here at one section of the 28S gene, and each row, just for clarification, is one individual tick. So we have the unknowns, and they're aligned with the published GenBank sequences for Exodus scapularis and Exodus cookii as reference points. These yellow boxes here are 2 of those markers that I was talking about. So based on the nucleotide or the letter at those markers, we can determine